Thank you for joining the Foundation for Sarcoidosis Research webinar on FSR funded fellows research update. The webinar will begin shortly. There will be silence, but please know that you are connected. Good afternoon. Welcome to the Foundation for Sarcoidosis Research's webinar on FSR funded fellows research update. I am Chase Hamilton, FSR's research coordinator. One item I wanted to address before we get started. Unfortunately, Dr. Kiyomo will not be able to attend the webinar today. We apologize to those who were looking forward to hearing her present. We will be circling back around with her to find an alternative time for her to share with you updates from her research. And just a quick bit of housekeeping. Your phone will be muted. However, we would like to encourage you to ask questions. There's a Q&A box located in the bottom of your screen. Type your questions into this box throughout the presentation. We have disabled the chat, so please use the Q&A box for questions. We will have a question and answer portion period at the end of this webinar. And this webinar will be recorded and will live on our website. Next slide, please. I'm now pleased to introduce Mary McGowan, FSR CEO, to kick us off today.
Thank you, Chase, and welcome everyone. We are thrilled to be joined today by those of you who share our passion for better treatments and a cure for sarcoidosis. FSR is the leading international nonprofit association committed to accelerating research in sarcoidosis, providing education and support to those living with sarcoidosis and their care partners, and raising awareness about sarcoidosis worldwide. Next slide, please. FSR was founded over 20 years ago. Since that time, FSR has provided over $5 million in support of sarcoidosis research. FSR is dedicated to providing education, support, and advocacy for patients living with sarcoidosis and the physicians who care for them. And FSR has over 60,000 members in our community. Next slide, please. So we have a four-pronged approach. First, we work to advance and accelerate research in sarcoidosis. FSR has robust fellowship and grant programs to support researchers in the sarcoidosis space. In addition, through the SARC Connect program, FSR works with academic and industry partners to identify willing clinical trial participants and to ensure that all research remains patient-centered. FSR also provides those living with sarcoidosis numerous support opportunities, including wellness programming, social events with others who have similar lived experiences, and one-on-one support through our Patient Navigator program. Additionally, FSR is committed to providing educational opportunities for patients and clinicians in order to provide them with the most up-to-date research and clinical care information in sarcoidosis. Finally, FSR advocates for researchers and clinicians and for those living with sarcoidosis by encouraging more funding for sarcoidosis research and working to reduce access challenges. Next slide, please. We are very excited to have closed out our successful Sarcoidosis Awareness Month. April's theme, Make It Visible, has resonated worldwide. Throughout this national campaign, we shared clinician stories, as you can see here from Dr. Elliot Krauser, FSR Scientific Advisory Board Chair, and also patient stories both in the US and actually internationally. Patients received proclamations from local governments to recognize that April was Sarcoidosis Awareness Month. Additionally, 2 of our hospital partners organized buildings to be lit up purple in their cities in honor of World Sarcoidosis Day and sarcoidosis awareness. We were thrilled to see the community's engagement with our Awareness Month t-shirt, which will help to raise awareness year-round. Next slide, please. Just a few weeks ago, FSR hosted a patient listening session to the FDA for pulmonary sarcoidosis. In this influential listening session, there were 6 patient speakers and 1 care partner. Dr. Lisa Meyer represented the FSR Scientific Advisory Board, providing analysis of this disease from the physician standpoint. There were 50 members from the FDA in attendance. Thank you to all who completed our survey. We used your responses in shaping the messages that we put forth to the FDA. I would like to draw your attention to the encouraging quote from the FDA Director of Pulmonary Division. FSR plans to put out a white paper, host a community webinar to present more information about this milestone event, and in the future, we will be hosting a patient-focused drug development meeting at the FDA. Next slide, please. We are so proud of the research that is coming out as a result of FSR-funded activity. Last year, there were a grand total of 26 publications released. This year, there are already 6 publications in addition to the many abstracts being released at the American Thoracic Society conference coming up later this month. Next slide, please. We have 2 exciting grant opportunities currently accepting applications. Our pilot grants fund new and promising studies in sarcoidosis that might not otherwise be funded and are used to help academic researchers advance a study for future funding. The Cardiac Sarcoidosis Grant is a brand new grant opportunity developed with FSR Scientific Advisory Board. This grant looks to fund research that will provide a deeper understanding of the causes, progression, or treatment options for cardiac sarcoidosis. Please share these opportunities with any researchers you believe would be interested. Next slide, please. And one final item that I would like to draw your attention to is our patient registry. There are 5,000 patients enrolled and 46 countries represented in our registry. This registry is a questionnaire to help physicians and researchers better understand your sarcoidosis journey, how sarcoidosis impacts your quality of life, and to better understand the burden of disease measures. We encourage you to join the FSR registry to help tell your story to physicians, academic partners, and industry partners to help develop better treatments, improve quality of life, improve diagnostic methods, and move closer to developing a cure for those who are living with this disease. I would now like to take this opportunity to thank the fellows for speaking today. We are excited to learn more and share with you progress that has been made by our 2018-2020 FSR fellows. I would like to now pass it back over to Chase, FSR's research coordinator, to provide you with an introduction to one of the FSR fellows speaking today. Thank you.
Thank you, Mary. I would now like to introduce FSR 2018-2020 Fellow, Dr. Brian Young from Yale University. Dr. Young received his MD and PhD from UT Southwestern Medical School in Dallas, Texas, completing his doctoral work in cardiac genetics in the laboratory of Dr. Eric Olson. He trained in internal medicine at Yale, where he remained for cardiology training in Yale's fellowship, directed by Edward Miller. He worked as a postdoctoral research fellow in the laboratory of Dr. Jeffrey Bender, studying inflammatory biology in the cardiovascular system. He is now a clinician in the Yale University section of cardiovascular medicine and the Nuclear Cardiology Center at Yale New Haven Hospital. His academic interests include basic, translational, and clinical research in cardiac imaging. He was a recipient of FSR's 2018-2020 Sarcoidosis Research Fellowship, which supported his efforts in developing better methods for monitoring cardiac sarcoidosis with PET imaging and specialized blood biomarker tests. More recently, he has provided specialized imaging to assess outcomes in clinical trials seeking new sarcoidosis therapies. Dr. Young has presented at 4 national meetings and published 4 papers with another publication in progress. His research group has been supported by the NIH, private industry, and the Veterans Healthcare Administration. I will now turn it over to Dr. Young.
Thank you, Chase. It's actually hard to believe it's been 4 years. I was just sitting here hearing 2018 and thinking, wow, that's it's amazing. It's really not necessarily been the 4 years that we all expected in some ways, but we've made a lot of progress, and I'm really excited to share that with you today. I want to first and foremost thank FSR and the sarcoidosis community for supporting me through this by way of this fellowship. It's a lot to review in just a few minutes because it is not only the 2 years of work that I did during the fellowship, but an additional 1.5 years since, in which we've really enjoyed the momentum that we gained early on from this early career support. So I'd like to give you a little context of where this fit into the early phase of my career, just to emphasize how important this was and how appreciative I am. Please go to the next slide. Next slide. So this is a little outline of how this worked, and I think it's really important to understand where this plays into the early career life of an academic researcher, and that's that this is the thing that supports us right out of training. In training, you have support through other mechanisms, through training grants and things like this, but it's that early faculty phase where we really just need that start before we have the big up and running lab, before we have the research coordinators and infrastructure that more advanced career researchers have. So I'm a clinical cardiologist at Yale. I work at Yale New Haven Health in the VA Healthcare Administration. And my interest is in treating cardiac sarcoidosis in those contexts, but I'm also interested in doing research that's grown out of the fellowship. So in 2018, I finished my clinical training, and my FSR fellowship started literally 48 hours after my training completed. Took a weekend off and hit the ground running thanks to the support. And not having that laboratory or research staff, it was so critical to have these resources, to acquire reagents, to acquire the time of some research technicians and study coordinators to get these projects off the ground. This was my first funding outside of the training grants for my training period. So I was able to get a lot of momentum early because of this, and collaborate both within Yale in the multidisciplinary clinical and research groups that we have already in place, as well as labs at other universities. About less than 6 months, in fact, after starting, we had multiple collaborators on these projects, each with their own funding and own expertise and own resources that they brought to the table. So we started the biomarkers project on day 1, and that's an effort to study blood tests for cardiac sarcoidosis. The clinical trial that I'll talk about just a little bit later was just beginning around the same time within the Yale Rheumatology group. And during that first year, I began working with them. Their funding was through the NIH and industry sponsorship, it being a clinical trial. And I personally would not have had the time and research infrastructure to contribute in any way to that effort if it were not for the pieces that I already had up and running in just a few months because of the support through FSR for our cardiac nuclear imaging facility and the fellowship. About a year in, I started making the scientific meeting circuit back when we used to travel to these meetings. And as the data started coming out of this project, I got invited to several meetings early on before we had published anything. And by 2020, we were able to get these out in the form of publications, both the biomarker data, the early results of the clinical trial, and several other reviews and supporting publications that come along as you start to put these things together. And then we saw a few more publications in late 2021. So I'll highlight a few aspects of our aims on the next slide. This is what we're seeking to do. And I'll actually start with the middle one, because that's the one we actually started first in time. And that is to find new, simpler tests. And any of our patients will tell you that if you have cardiac involvement in sarcoidosis, it's a difficult path. It's a difficult path because we have to treat cardiac sarcoidosis even when it is asymptomatic. And that means we need a really good test to detect a thing that you might not feel, but it is important to treat because the outcomes can be so bad if it develops undetected without being found. So one of the things that has been on the wish list for cardiac and other types of physicians that treat sarcoidosis for a long time are biomarkers that allow for early detection of disease and, most importantly, fewer scans. So in the next slide, I'll show you why that's important. This is the current treatment paradigm for how we deal with cardiac sarcoidosis, where I and others in cardiology clinic evaluate patients that typically have been referred to us from other physicians, especially pulmonologists. And these patients either have known cardiac involvement of their sarcoidosis or their physician suspects that they may have cardiac involvement because of some symptom they've had or something that's been seen on some other imaging. So in addition to common tools like EKGs and echocardiograms, two of the major tools we use are cardiac MRI and cardiac PET scans. And I can't go into all the details of how exactly these work in a talk of this length, but they are challenging. They're difficult scans, both for us as physicians, but more importantly for our patients to endure. These are long scans. They take most of a day. In the case of the PET scan, they take a lot of preparation the day before. And when you don't have anything else, when you don't have a biomarker, you don't necessarily have easy things like EKGs and echocardiograms telling you how the disease is doing, a lot of these very difficult, very expensive radiation exposing scans have to be done serially, meaning repeatedly to know how disease is doing and whether treatments are being effective. So what we like to do is insert a simple blood test somewhere in this treatment paradigm. So even if it doesn't take the place of the scans completely to guide us as to whether to go up or down on therapy or add new therapies, it at least can give us a clue as to when we might need to do imaging versus just watch and wait and continue to follow clinically. So on the next slide, I'll show you schematically how we approach this. So our first aim when I started fellowship was to start collecting blood samples, very special research blood samples that had to be processed in a very certain way when the patient came in for PET imaging to diagnose or monitor their cardiac sarcoidosis. And that enabled us to create a collection of samples categorized by whether the patient had active disease at the time the sample was taken. This is a little different than biomarker studies in the past, for which we've had several decades of experience. Other studies would just take blood from patients without sarcoidosis, kind of like normal, healthy volunteers, and then they would take patients with sarcoidosis who showed up in clinic and they would ask them if they'd be willing to volunteer to give blood. And we can compare the blood from people who do and don't have disease, but that didn't really tell us what we needed to know, especially as cardiologists, and that is whether the disease in a patient who's already been diagnosed, whether that disease is active now. So what we did is we took the blood at the time of the PET scan, and that enables us to say not just yes sarcoidosis or no sarcoidosis. That enables us to say this person has active disease now, or this person, even though they have the diagnosis, doesn't have any active disease now, which is what we as the doctor needed to know in clinic to say you need more prednisone, you need less prednisone, you need a second drug, you need a third drug, or we can dial back on those second drugs. A lot of these drugs are hard to take, and we don't want to have our patients taking them more than they have to. So on the next slide, I'll show you how these results integrated with our treatment protocol. So we got these tests, we put them in a biobank, in the Yale Biobank, which is done in part in collaboration with our colleagues in pulmonary critical care. who see more of these patients than we do. And we got these samples. We did a large array of tests on them using some specialized approaches that was developed by one of our collaborators. And we were able to find various biomarkers, which are simply blood tests that look for proteins in certain substances in the blood. And what we found was that there were biomarkers that correlate specifically with whether there was active disease in the heart. And so we're hoping someday that some of these findings, which tell us a little bit about the biology of sarcoidosis as well, will be able to integrate that into this care pathway and reduce the number of PET scans and MRIs our patients have to endure. And that brings me to the next slide, which is that these early results that we got in the biomarker tests enabled us to kind of get a footprint in the field so that we were recognized as people who had a new and accurate way to measure the success of treatment. And so we had always been doing PET scans to do this, but as we started to get those publications out, we started to be contacted by other researchers who were interested in collaborating with our PET center because we had expertise both in the research space and the clinical space of looking at these patients and saying quantitatively, meaning to really put a number to it as to how much active disease these patients had in their heart and whether it was getting better or worse. And so when you have a measure of success, that's useful clinically for your patients when you're making clinical decisions. But the other place that's useful is in clinical trials when you're trying to find new treatments for the disease. And part of the reason we don't have a lot of data for these therapies that we often use is because the outcomes are hard to measure. You don't necessarily want to put a patient on a new drug not knowing whether they're going to have heart failure or a dangerous life-threatening heart arrhythmia. You want what we call a surrogate marker. You want something that can say this disease is getting better or worse before the patient has a meaningful bad outcome that affects their life. So having this PET scan technology refined for our own studies, we were approached by researchers within Yale University, actually in the Department of Dermatology. And on the next slide, I'll show you the huge multidisciplinary group that it took, one of whom is presenting later, but also the people on the top there, Brett King and Bill Damsky, are dermatologists. And we in cardiology, we don't work with dermatologists a whole lot, to be honest. But in this case, they had a study going and they came to us and said, you know, we're trying this new drug. This is an FDA drug that's approved for other diseases, but it's an off-label for sarcoidosis, like everything else we use. And we're trying to evaluate this drug, and Pfizer is helping us support us, and the NIH is helping support us. But what we don't have is a way to quantitatively measure the success of our treatment. And so they had patients on this drug, and besides looking at their skin and measuring it in other ways by biopsies, what they wanted to add was some kind of imaging that would really put a fine number to it. And when we do imaging of the skin, what we're doing is the same PET scan we would do for the heart, which means we get data about these patients who are coming in for their skin, but we get information as well about what's going on in their lungs and their heart. And that enabled us to find things that we weren't necessarily even looking for. And on the next slide, I'll show you what I mean by that. This is a patient who had refractory cutaneous sarcoidosis. That means sarcoidosis of the skin that just was not responding to any conventional therapy that had been used in the past. And on the day before treatment, on the far left-hand side of the slide, if you look down on the patient's right leg there to the bottom of the screen near the knee, you see all that black patchy stuff. That's actually the sarcoidosis disease of her skin. And that completely resolved in 6 months of treatment on this unapproved therapy that's still under investigation. But what was interesting is that in the same patient, this was a patient who didn't even have symptomatic disease to know that they had sarcoidosis in their lung. And what we found is that they did have disease in their lung, and that too resolved with therapy. And we were able to adequately track that with the PET scan, like we would any other lung sarcoidosis patient. And we're able to quantify that and monitor that and showed that that was another aspect of this treatment that was useful. And thus potentially, hopefully someday, expand the indications of the drug. On the next slide, I have a couple more examples of this, where two different patients had disease in these cases, they had a lot of disease in the lung, and it resolved with a few months of therapy. We actually did our follow-up at 6 months in these cases. And the other thing that you see, if you really look closely, believe it or not, that patient on the left, those two images, that before and after, that's the same guy. And I want you to notice, if you look at the hazy outline of his body, is that he was a lot bigger 6 months. He got a lot smaller in the 6 months of the trial. And the reason for that is because on therapy, he was able to go off prednisone. And a lot of our patients will know that one of the very difficult side effects of being on prednisone, especially being on prednisone a long time at high doses, is difficulty with weight, difficulty with blood glucose control and diabetes. So this is just another example of how, if we find new therapies, we could integrate that with our existing therapies and improve the side effect profile of our overall treatment. So I was happy to be a part of this study, which again was an off-label use for an existing drug that was supported through pharma and through the NIH, but something that I never would have had anything to bring to the table for and add to this work if it not been for the sarcoidosis fellowship that protected, that facilitated my spending time and developing the infrastructure to do these kind of imaging studies. So that brings us to today on the next slide. And that is, today we're working on better imaging protocols within our PET center. And I want to discuss just the next big milestone, a project that we're just about to submit to publication. I just had a meeting with our collaborators in Beijing last week to try to put this together. The publication itself is going to be quite technical in nature. It's not going to be a fun read, but the result is that we're introducing a new way to do the computer processing of our cardiac PET imaging that will improve its accuracy. And this project would not have been possible without some of our patients being willing to stay for extra scans after their clinical scans, so that we can get images at 2 time points that are then kind of processed together with a big computer to derive special new images that enable us to look at the heart in a new way. It's called dynamic PET imaging. And that actually brings me to my last slide, which is the most important thing, which is to thank our patients first and foremost, who've been willing to give us blood, give us time, give us extra scans that were not even part of what they needed clinically, but just because of their willingness to really give of themselves as well to contribute to this research, we were able to develop this huge biobank. We're able to develop this huge library of imaging studies. We're able to use an experimental drug with experimental scans. And many of these patients are now coming in for their 2nd and 3rd scan now, more than 2 years now after the trial has started. So first and foremost, to our patients, thank you for participating with us and making this research possible and continuing to do so as we enroll more patients now as the pandemic dust settles. Thank you to the Foundation for Sarcoidosis Research for supporting me. It couldn't have been earlier in my career. As I said, it was literally day one that this support enabled me to do this research right out of training, so that we can care for our patients, but more importantly, make all that testing and treatment for them easier in the future, to make the testing more reliable, more effective, to guide therapy. And I think I'm going to pass it back at this point to Chase.
Thank you, Dr. Young, for that great presentation and update. We really appreciate it. I will take the time now to introduce Dr. Ryu from Yale University. Dr. Ryu is an assistant professor of medicine in the section of pulmonary critical care and sleep medicine at Yale. He received his BS at Emory University, MPH at Dartmouth College, and his MD at New York Medical College. After completing his internal medicine residency, including a year as chief resident at SUNY Upstate Medical University, he came to Yale for his pulmonary critical care fellowship. During his time as a clinical fellow, he joined the laboratory of Dr. Erica Herzog, and under her mentorship, he has conducted translational research in idiopathic pulmonary fibrosis and sarcoidosis. His research involves the study of pathogenic mechanisms by which extracellular mitochondrial DNA contributes to disease pathology and clinical phenotypes of idiopathic pulmonary fibrosis and sarcoidosis. Along with funding from the FSR, he has received grant awards from the Parker B. Francis Foundation, Boehringer Ingelheim, Chest Foundation, and most recently an NIH K Grant Award. While his K grant will focus on idiopathic pulmonary fibrosis, he has continued his work in sarcoidosis with a Chest Foundation grant to explore the pathology of stage 4 sarcoidosis. I will now turn it over to Dr. Ayub.
Thank you so much, Chase, for that kind introduction. And thank you all for having me here. Definitely enjoy speaking with the FSR community about my research experiences, and also just as a fellow with the FSR. And so Dr. Young really highlighted a lot of the challenges that come about in the early career stage of faculty, especially in doing research. And I was in a similar position as Brian was. I just graduated fellowship, and at that time you do need research funding to help continue your research platform going. And so the FSR, as I'll get to in a second, was one of the first career development awards that I've had, and that really was the springboard to me developing my research platform. Next slide, please. Next slide again. Thank you. And so, so all I can say that is, yeah, just echo the sentiments that Brian mentioned, that I highly recommend the FSR fellowship to any early stage investigator, especially with the interest in sarcoidosis. And I would just say, to sum it up, it really empowers, the funding from the FSR really does empower you as a scientist, not only to study the work that you're doing, but also to expand your research platform to other collaborators. You know, I did work with Brian on his research project, and you know, I don't really know anything about PET imaging of cardiac sarcoidosis, but this gave me the opportunity to do so. And I'll get to some other projects I've been fortunate to be a part of, and much of this was facilitated by the FSR. And so, again, highly recommend to all early stage sarcoidosis investigators. It's also been just enlightening to learn from members of the FSR community in these types of settings, these online webinars. I think just hearing other people's work has just been amazing, as well as just in conferences. You can really see the cutting edge of research that's being done. The community is also a little bit smaller, and so what I've also found interesting is that helpful is actually just to engage with some of the thought leaders in the sarcoidosis field. Dr. Krauser, Dr. Wonder Drake, are just two amazing scientists, and just haven't been able to meet them in some of these conference settings through through FSR sponsored events has been really helpful. And then, just like Brian said, this has really supported my career as a physician scientist. It was the first career development award. And what that means is that the grant will provide 75% protected research time to do research, and where the remaining 25% of my time is seeing patients in the sarcoidosis clinic. And so having that protected time in my career really was helpful in getting, completing my research projects, and then ultimately getting an NIH K08 award, which gives me 5 years of funding with 75% protected time. And then the last thing I do want to say about being part of the FSR community is that it has tons of opportunities for collaboration. I'm a translational scientist, meaning that I do research on the laboratory bench, but it's using samples from human patients. And so a lot of this work, a lot of my work is dependent upon the existence of biorepositories. And many of the patients here may have may be familiar with the GRAD study that where we collected samples from bronchoscopies and blood from about 2012 to 2016. I myself have utilized the rich resources of the grads cohort. I've also used resources from the biorepository from Access Sarcoidosis, which was one of the first ones. That spanned across another 5 years from the late 90s to the early 2000s across 10 different sites. And so these opportunities for collaboration are really invaluable for me as a translational scientist. Next slide, please. And so I do want to thank everyone at the FSR organization, particularly for the community engagement has been amazing. These online webinars, the newsletters that we get are also have been helpful in making sure that everyone in the sarcoidosis community is aware of what's going on and together. I do also want to say that the social media, especially the FSR Twitter account, has been a great follow. And so just managing that on a day-to-day basis has really helped me really stay abreast of what's going on and related to sarcoidosis and also in the lung disease field. I do want to shout out my FSR mentoring team. Dr. Erica Herzog is my mentor. She's done work for the FSR and published with me in our sarcoidosis research. Murdu Galati runs the our sarcoidosis clinic and has really done all the legwork to get it to be recognized by the Wasatch Association. And then our section chief is Neftali Kaminsky, who's a renowned investigator in interstitial lung disease. And he's actually, it was actually him who really showed me that sarcoidosis is an underrepresented, understudied field and encouraged me to go into this, down this path. And then, of course, all of our patients who are here listening with us, who've participated in clinical trials and participated in biorepositories, have really made all of this possible. Next slide. And so briefly, I'm going to just talk about my FSR grant and the work that we were able to accomplish and some of the next steps that we've done. And I'll walk everyone through some of the through the scientific terms here. But what we found, other people have found before, is that when cells undergo stress, they will release their mitochondrial DNA into the extracellular space. And in that case, it could be in the plasma or in the serum. And so we have found that high levels of this mitochondrial DNA in the plasma was actually associated with poor outcomes in another lung disease, idiopathic pulmonary fibrosis. And we found that this mitochondrial DNA will actually bind to a receptor known as toll-like receptor 9, TLR9, which then triggers your immune system. And as a result of that immune activation, it leads to the production of these different cytokines that then cause inflammation and fibrosis. And because sarcoidosis shares some of these mechanistic pathways with IPF, we believe that there's a possibility that there's a link between mitochondrial DNA and clinical outcomes in sarcoidosis. And so our first aim was to identify whether identify the presence of this mitochondrial DNA in sarcoidosis, particularly among African American patients who we've known for quite some time have always had poor outcomes with sarcoidosis. And we're looking to see if there was a biologic basis through this mediated by the circulating mitochondrial DNA. Next slide, please. And so again, as a translational scientist, I leverage access to two sarcoidosis cohorts. We have our local cohort of patients at Yale, and then we had the grads cohort, which was from 2013 to 2016 across 9 different institutions, and we had about 270 or so serum samples And along with these serum samples, we had, if any of the patients who participated knows, that it includes different questionnaires, pulmonary function testing, results of their CAT scan, just to name a few items. And then within these serum samples, we actually measured their mitochondrial DNA levels, and then we also measured the ability of that plasma to activate toll-like receptor 9, TLR9. And then we worked to see if there was a relationship between mitochondrial DNA and TLR9 activation and clinical outcomes of sarcoidosis. Next slide, please. And so this work, which we published a couple years ago now, we found that these mitochondrial DNA concentrations were elevated in the plasma of both very elevated, actually, in both sarcoidosis cohorts, and that this mitochondrial DNA was associated with TLR9 activation. If you look on the middle over there. And so that was exciting for us to see that finding, that maybe this mitochondrial DNA TLR9 activation is what this suggested to us, is that maybe it led to the production of different cytokines that then elicited inflammation and maybe fibrosis. And so the other aspect of this project is that we also broke down our patient cohorts between race, between Caucasian and African American patients. And what we found was that African Americans had higher levels of mitochondrial DNA in their plasma. And specifically, we found that African American patients with extrapulmonary sarcoidosis, meaning they had evidence of sarcoidosis anywhere outside the lung, had especially higher levels of mitochondrial DNA. And so to put this all together, we found that just having plasma mitochondrial DNA, high levels of plasma mitochondrial DNA, put you at increased risk of having extrapulmonary sarcoidosis. African American race also independently gave you higher odds of extrapulmonary sarcoidosis, and that these odds went up again if you were an African American person with high plasma mitochondrial DNA. And so we looked in, and the reason why this is important is that when we see patients in clinic, particularly a newly diagnosed person with sarcoidosis, we want to know what is the possibility that there's disease beyond the lung. And as some of the patients know, we don't want to put you through unnecessarily testing if we don't have to. And so maybe measuring mitochondrial DNA is one clue or one insight into whether or not there's disease outside of the lung. The other part that we also wanted to look into is really looking at this relationship between mitochondrial DNA and TLR9, and the best way to study this. Because if you have high levels of mitochondrial DNA, our data suggests that your immune system is chronically activated through TLR9, and can we leverage that knowledge to ultimately develop better therapies beyond the ones that we have in existence today. Next slide. And so the next step here is really to study this relationship through developing models of sarcoidosis that we can study in the lab. And so one approach that we have in the lab that my mentor, Dr. Herzog, really has put on the map and was really an expert in is developing models of lung models using a detergent-based method, where we take lungs that have been explanted from patients who are undergoing lung transplant or who have passed away, and you create these scaffolds where we can create these scaffolds that have the biochemical and biophysical cues of the diseased lung, and that helps us model model the sarcoidosis environment. And then there we can expose cells to different conditions and maybe identify a way of better understanding mechanistically what's going on between TLR9, between mitochondrial DNA and TLR9. Next slide. And then the other direction that we've gone to, and this is where I was talking about between having the opportunity to look at different projects, especially by having a funding mechanism like the FSR, is to look at other aspects of other projects that study different areas of disease. And so as someone who's interested in pulmonary fibrosis, stage 4 sarcoidosis is also is a deadly condition where, is a deadly condition where there's scarring of the lungs for which we do not have good treatments. And so we do know that the development of pulmonary fibrosis, scarring the lungs, is a result of an imbalance between high collagen formation and low collagen degradation. And when there's that imbalance, we do see that causing, that leading to the development of lung scarring. And so we can measure, these products of collagen formation and degradation. And perhaps that would be one way for, and if we, by doing so, it gives us a little bit of additional insight into the lung, into the fibrotic lung, and maybe gives us different treatment strategies or tailor our treatment strategies based on our knowledge of whether or not there is whether or not there's an imbalance in this collagen turnover. Next slide. And so what we found was that in 2 cohorts of sarcoidosis patients, both from one from Access and one from GRADS, that there is actually high levels of all these collagen, what we call collagen epitopes, collagen markers in their serum. And so our project now is to look into what's going on here. Is this too much? Is there too much collagen? Is not enough collagen being degraded? And whether or not and the pathogenic potential of these collagens, of collagens in terms of, is it causing pulmonary fibrosis? Is it causing extrapulmonary disease? And again, by understanding what's going on with collagen turnover, we might be able to develop better ways of treating this condition. And so with that, that's actually my last slide. And again, I just want to thank the FSR for having me give this talk. And I think now I'll hand it off to Chase, or I guess Mary.
Yeah. Well, thank you so much, Dr. Rio, for that interesting presentation and update. And so interesting, the variation in race outcomes. Thank you for highlighting that. And thank you also to Dr. Young for the informative presentation on your extraordinary work on more effective imaging. At FSR, we understand these young researchers are doing critically important work, and we are so very proud of their successes. We wanted to give these fellows a chance to share their successes directly with you, the sarcoidosis community. Through the dedication of our community, the careers of these dedicated researchers to drive progress in the advancement toward more timely diagnosis, treatments, and a cure is an opportunity. Now, we would like to provide you the opportunity to ask our fellows any questions that you have about their research. As a reminder, please type your questions into the Q&A section located at the bottom of your screen. Thank you.
Thank you, Mary. So to begin, I will start with Dr. Young. Dr. Young, can multi-system sarcoidosis eventually also affect the heart? As a follow-up question, how often should patients be checked for active sarcoidosis in new areas?
Both very good questions. Yeah, the pattern of organ involvement is highly variable. among our patients, with the most common organ being affected being the lung. And what that means for a lot of our patients in our center, and I think most centers, what happens is because the lung is so frequently involved, that becomes the pulmonologist becomes the sort of quarterback of the team, directing a multidisciplinary team that might involve cardiologists and neurologists and dermatologists, as things develop. So I would say that nothing is never or always in sarcoidosis, meaning that you can have multi-organ involvement and never see any cardiac involvement, which is actually among all sarcoidosis patients. If you take everybody who has involvement in their lung, involvement of the heart is actually quite rare overall. But because the outcomes can potentially be so bad, we're very, very careful about it. So the way to approach this with your doctors is just to have a pulmonologist who really is comfortable treating sarcoidosis, with some experience doing so. It's not that common in the big picture, compared to other things we treat, like asthma and COPD and coronary disease. It's a rare disease, so you need somebody who knows how to direct that team and can pick up on the signs clinically, when to refer to somebody else outside of their discipline. So remember that a lung doctor was an internal medicine doctor first, just like a cardiologist was an internal medicine doctor first, and that they'll be able to help you and guide you, and know when you need a referral to a specialist in another field. Most people, fortunately, most patients get away with not ever having to meet me, and ever having to have a PET scan. And it usually starts with the cardiac MRI, actually. And if the cardiac MRI is normal, then in most cases we're able to avoid my PET center. But it's a good question. When you have multi-organ involvement, it's important to really keep up with your doctor, really follow up regularly, and really have somebody who's comfortable working with the team.
Thank you, Dr.
And the other,
Young.
there was a second question too, right?
Yeah, so there was a follow-up question. It said, how often should patients be checked for active sarcoidosis in new areas? And then, an additional question would be, what are the benefits for patients to get PET scans, and what can be the restrictions of getting those PET scans?
Yeah. The follow-up just depends on the clinical circumstances and how active your disease is. So that's why you should, in most cases, you're going to be seeing a pulmonologist regularly for lung disease, or a dermatologist for dermatologic disease. And in those contexts, those doctors should be looking for other signs of other involvement and referring you accordingly. It's usually in terms of some number of months, is when we typically see our patients on a kind of a rotating basis. And then the barriers. So the PET scan, I didn't have time to talk about a lot of what goes into that. It's a very tough scan, as any patient who's gone through it will know. And it involves a dietary prep that goes for a full 24 hours before you get your test. And then you have a test, which takes up almost all of your morning. It's a couple scans, there's radiation exposure, there's, laying in a scanner that not all people are comfortable with. You have to get an IV. It's a 36-hour process from the beginning of the prep to getting out of our center. So, more or less. And so it's not something we want to do a lot. And unfortunately, in a lot of cases, it's something we have to do on a repeat basis. So the barriers are, one, finding a center that's experienced with it. A lot of centers have PET scanners because it's used to monitor cancer and things like that. Not every center can do a cardiac PET scan for sarcoidosis. This is a pretty specialized thing. So to get one of these things, to get one of these scans, you're going to have to be in probably a major academic medical center. You're going to have to be in some kind of institution with university in its name in order to find somebody who knows what they're doing with these, both the cardiac MRI or the PET scan. And the advantages are that we can monitor disease that's not symptomatic, meaning disease that you don't know you have. We can find it in the heart when there's suspicion that it's there based on other tests like EKGs or symptoms or echocardiograms. We can find it in the heart when it's maybe hasn't done, hopefully, much damage, and we can treat it and cool off the inflammation with drugs so that it never becomes an abnormal heart rhythm or heart failure or something that is symptomatic. So it's a very sensitive tool. It's hard to use, it's expensive, it's hard to find, but we use it when we need to use it.
Thank you, Dr. Young, for that insightful response there. I'm going to shift over to Dr. Ryu. I have a question here. Are there any non-steroidal inhaler treatment that can help manage pulmonary sarcoidosis?
Right now, we don't have anything that is non-steroidal that would help with inhalers that would help alleviate pulmonary sarcoidosis. We do have some one in development right now. It's actually in clinical trial for IPF, but stuff that we do in IPF ultimately does we do end up trying to study that in sarcoidosis. So presently not now, but hopefully in the future there will be.
Yes, hopefully in the future. Yes, we will look forward for that. And then this question is for both of you guys. I'll have Dr. Yu answer first here. How can patients encourage their pulmonary and rheumatologists to take an active part in sarcoidosis research?
Yeah, so I think that as Dr. Young alluded to, being in an academic center that's participated in clinical trials, period, um, is helpful because these places like Yale, for example, we are active in the clinical trial space. We're active in the biorepository space, and so we're always, if there's any studies open, we're always recruiting, and we will, try to help. We'll try to get that out there as much as possible. I think also it just helps to just be, a lot of people just don't know as well. I think you guys FSR have done a nice job of tweeting that out. Actually, I didn't know that Life Pharmaceutical was actively enrolling in a clinical trial until actually I saw it on a Twitter feed from you guys. And so we did bring this up with I did bring this up with Dr. Galati and other people to whether or not we should try to get Yale as a clinical trial site. And so I think, yeah, check in. I would just say check in with, if your lung doctor or rheumatologist is not affiliated with the university center, see what's going on. Reach out to your major university center and see what they can do. And I would imagine if there is a trial, we're always trying to recruit and enroll patients whenever possible.
Thank you, Dr. Yu. Dr. Young, do you have any additional thoughts for that response?
Yeah, I think it's in addition to what you said about academic medical centers, it's really important that we and what FSR's work does in reaching out to clinicians as well, because we can enroll patients in trials and we can involve people in clinical research, outside of the big medical centers as well, from providers who are interested in assisting their patients find these clinical trials. So it's like you said, it's Twitter, it's FSR's website, it's just awareness that there are ways that patients can participate and that physicians can participate even if you're not in the academy. And FSR is really a key component of all of that by consolidating that information, being a repository of these talks.
Thank you, Dr. Young. So we are running a little short on time. I just want to ask one question to both of you moving forward. We can have Dr. Young go first here. How has the FSR Fellowship Grant helped to contribute to your research?
It started it all. It's hard. It's difficult to get started. When you are a well-running research operation with preliminary data and momentum and a track record and a lot of papers and staff, I want to say it's easy to get funding, but it's a lot easier. It's really hard starting out right out of training to really get your footing and to not have a freezer to put samples in and not have a research technician to do work, and to not have any of that is a major barrier to people coming out of training and doing research at all. Because you can come out of training and just be a pulmonologist or just be a cardiologist. And when you have somebody who's willing to kind of take you on faith and give you a grant based on what you propose to do and just a feel for the fact that you're passionate about doing it, it lets you get started, and it lets you apply for other grants, and it lets you get the data you need to get out there and meet people and make connections and collaborations that lead to more grants down the line and in mid-career.
Thank you, Dr. Young. We really appreciate all the efforts you've made for FSR here, and I'm going to pitch it over to Dr. Ryu to give him a chance to respond to this question.
Yeah, I definitely echo all the sentiments that Brian just mentioned. Yeah, I would say that what's really also helped me is it just really helped me pursue different collaborations. So I got to work in that in the sarcoidosis space with a bunch of different with different collaborators from different fields, and so I think that's certainly been incredibly helpful. One publication in sarcoidosis I haven't really mentioned is that I was looking at this this cytokine, this immunologic, this mediator of our immune system. All the data was negative actually, but we're still able to publish it. But there's really not much I can really go with that. But that was a risk that I was able to take. Because it was just because of the expertise that we had here at Yale. We did not find a signal in anything, but just to be able to do something like that, it would have sunk a lot of other people's careers if they didn't have the funding. But I got to really get to look into the science behind what's going on here and pursue different things. Look and really study this and delve into it, and it's a real opportunity that most people unfortunately don't get. So I'm certainly grateful for it.
Yep. Thank you, Dr. Ryu. We really appreciate you guys taking the time today to present and give our community an update on all the research that you have completed, and give us some background on what you're moving forward with in the future here. So if I could have the slides back up, there are just a couple of pieces of information that I want to cover before we end the webinar here. But I want to take this time to thank you for joining today's webinar. Thank you to the FSR Fellows for the updates and their continued hard work in the field of sarcoidosis. If you enjoyed today's event, we hope you will consider donating to help us continue to support young and upcoming researchers in the sarcoidosis field. These events are only possible because of our generous community. To receive updates on FSR research and learn more about educational opportunities like this, join the FSR community at the link you see on the page. If you're interested in participating in clinical research, please visit the link on the page to see what clinical trial opportunities are available. If you would like to hear more from researchers, attend the FSR Patient Summit, July 30th to 31st. And please complete the evaluation survey that will be sent to you via email after the conclusion of this webinar. The survey includes questions directly for you from our researchers who presented today. Next slide, please. And if you have any additional questions about our fellowships or grant programs, please reach out to me at the contact information that you see on the screen. Thank you for joining us today.
Thank you, Dr. Rio. Thank you, Dr. Young. I just want to end by reading something that was in the chat from one of the participants, and it's directed to you both. It says, "Thank you for researching these areas. So much of us are affected, and sharing your research with multiple explanation points. We appreciate you," all capitalized with an exclamation point. "You make such a difference for people living with sarcoidosis. Our hats are off to you for your extraordinary research. It's an honor to collaborate with you, and thank you for all that you do and for being with us here today." Bye-bye.
Great. Bye. Thank you for having me. Appreciate it again.
Thank you.